it's my pleasure to introduce my faculty colleague, Dr. Ted Kalbfleisch, as today's seminar speaker. Ted got his bachelor's degree at the University of Louisville in chemistry, went on to Boston University where he got a PhD in physical chemistry, my favorite topic. Then he did a postdoctoral fellowship at Boston University in the area of chemical physics. Ted then spent about 10 years in the pharmaceutical industry before landing back at the University of Louisville, first as the director of bioinformatics operations, and then as an assistant professor and associate professor in the Department of Biochemistry and Molecular Genetics. So we were able to get Ted to come here in about 2019 and join our faculty. We're delighted to have him because of his expertise in the area of domestic animal genomics. He's worked in the area of cattle and sheep genomics, but the real reason he's here at the Gluck Center is because he's been one of those key figures in the equine genomics efforts. And indeed, Ted was the lead author on the recent publication and release of ECCAB3, the new and improved reference genome for the domestic horse. And today, Ted's going to tell us about why there are zebra genes in your horse's DNA and what the implications of that might be. Ted.
Thank you very much, Dan. It's exciting to be here with you all today, and I very much hope you enjoy the contents of this presentation. So I've been asked to remind people who are looking for CE credits. You need to sign into the chat room and let folks know that you are here. Okay, so I'm going to so introgression may be a concept that is foreign to many of you. I was talking with a colleague who said they saw the title and they immediately had to Google it. So for those of you who haven't gone that extra step, I'll give you an introduction to that concept. In an evolutionary context, if we look how evolution acts at the genetic level, which is where evolution principally acts. Let's imagine a scenario where we have a sire and a dam, each possessing a maternal copy of a set of chromosomes and a paternal copy of a set of chromosomes. And both the maternal and the paternal copies of the sire and the dam are equivalent to the base across a certain region. And then they have a foal, and that foal has an allele, in this case a C, that cannot be attributed to either the sire or the dam. Now, misattributed paternity is not the culprit here. This happens a handful of times, tens to hundreds of times in every new organism that is conceived. Right? That a handful of new genetic variants simply happen. In almost all cases, these things are perfectly benign, but in some cases, they are good. And when they are good, what evolution will do is so if it is good, if it confers an advantage, then that animal will ultimately have an advantage breeding, and that allele will be passed on preferably to a point where in between 100,000 and maybe 1 million years, that allele can become fixed in the species, right? So new allele shows up, confers a strong advantage. The species as a whole ultimately accepts and promotes that allele through interbreeding, and that allele becomes fixed and it becomes a species-specific allele. Great process. It's worked very well for millennia, for millions and millions of years. The problem is it's slow, right? If something were to happen, if you have a change in an environment that would require adaptation at the genetic level for survival, this mechanism is likely not to help you. So is there another way that, that this can be accomplished? Well, we know in the wild and even on our farms that it's possible to take two animals from closely related species and cross them and produce an F1 cross. And in certain species, these F1 crosses will be fertile. The males typically aren't. The females are usually the ones that are. And then when that animal is bred to other animals, it will spread the genes from species A hither and yon to progeny in species B. So this is, if it's your intention to do so, it is a remarkably fast way to introduce a whole bunch of new alleles into a species. Okay? So in horses, we know that what we can do is we can cross a horse with, say, a donkey. That part works just fine. What doesn't work so well is that animal that mule that would be produced. It can't breed. There are no fertile offspring, so it can't pass genes on. Right. So how can this happen in horses? And that's something that I'll talk about briefly a little bit later. Okay. So why doesn't it happen in horses? The explanation that I've heard most often is that of chromosomal plasticity. Horses have 32 chromosomes, and donkeys and zebras can have in the 20s to in the teens of chromosomes, just making it very, very difficult for the reproductive machinery that is mitosis and such to function to produce fertile offspring. So it's a challenge, but what we don't know is actually when all of these chromosomes, when all of these chromosomal differences appeared, right? And these are just extant equids, right? There may be extinct animals that had very different numbers of chromosomes, very similar numbers of chromosomes to horses, such that they could in fact produce fertile offspring and produce something along the lines of a mule that could then be bred with other horses and retain the genomic material of that other animal. So yes, it's absolutely possible. These introgression events, when we talk about them, are typically hundreds of thousands to millions of years ago, and the landscape, the species-wide landscape, what existed, what the chromosomal structures were back then, is really anybody's guess. What species do we know that can productively interbreed? In Boss, cattle, bison, and yak all have 29 pairs of chromosomes. They can be interbred. Canids, the domestic dogs, coyotes, wolves all have 36. And cats, everything but leopards has 18 pairs of chromosomes plus a sex chromosome. And just as an aside, if you catch me in the hall and you want to learn about a really, really, really great way to build reference quality genomes, phased reference quality genomes, you want to do it with an F1 cross. That was a project that we worked on, and with some researchers at the USDA and the University of Nebraska-Lincoln, and we created from an F1 cross between a Highland bull and a yak cow. We produced two phased reference genomes, one for cattle, one for yak, both of which were more contiguous and more complete than anything that had been generated to date anywhere. It works remarkably well, and it's really cool. But it's a rather lengthy explanation. Okay, why would you cross animals? Well, for all sorts of good reasons. Among cattle, yak, and bison, you might want to augment traits of temperament, carcass traits, meat quality, coat color, things of that nature. Within horses, you cross a horse with a donkey, you get a mule as a beast of burden. A mule has advantages over a horse. In the wild, why would this happen? Well, there's this concept of adaptive introgression, and I hinted at it just a second ago. Okay, imagine that you have two closely related species pressed up against one another in one geographical region. And the environment begins to change in a way that one of those two species isn't well prepared to handle, right? It can't evolve, it's genetically, it can't evolve fast enough to deal with it, to deal with the problem. But if the other species is evolved and has all of these well-adapted genes, if those two are able to interbreed, you can bring over an entire, a whole genome's worth of well-adapted alleles in one fell swoop. And then when that animal breeds with other animals, it can selectively identify the genes that it needs to thrive in the new environment, okay? And when we speak about introgression events, we typically think of, oh, okay, one animal met with another animal and they produced a third animal, and that was it. Well, if you think about it, right, so what in evolution, right, you have an allele that shows up in one animal, and that allele ultimately has to get propagated over an entire species. Again, very slow. But if you have a maladapted species and a well-adapted species, every sire in that well-adapted species has all of the alleles that that other species needs to be able to adapt well, right? So you could have many events, many sires crossing with many mares from the unadapted species, and quickly bring in a lot of important genes for the benefit of that other species. So adaptive integration is appears to be an important mechanism to turbocharge environmental adaptation when evolution is just too slow. Okay, so I'm going to give you a wonderful example. I'm going to go through this quickly. There's this gene, EPAS1. Okay, if you're working with students, or if you all are students who are someday working with students, and you want to tell a really cool story about both convergent evolution and adaptive integration, EPAS1 is your gene. Look no further. Okay, so EPAS1, what is it? Well, when it's transcribed, it becomes this protein, HIF2 alpha. In hypoxic conditions, so when there's little oxygen, typically at higher altitudes, what will happen is cellular processes will begin to fail, and this HIF2 alpha dimer will be able to go into the nucleus and modulate the hypoxic response that a cell would have. Now, this hypoxic response is a pathway that's effectively conserved across metazoans. All of these animals have it. All these organisms have it. And essentially when oxygen gets low, what happens is you get a response that will begin to create more red blood cells, will begin angiogenesis, etc., which is a really good thing if this hypoxic condition is quick. And you go there, then you retreat from it, and then everything's fine. But if you're living in a hypoxic environment, then it's a real challenge. These responses can be detrimental over time, right? What's happened is in many species, they've done GWAS studies in animals of a particular species that have adapted to altitude versus those that typically live at low-lying altitudes. Consistently, what happens is you get a GWAS signal at EPAS-1, right? All of these species, they're different mutations, right? So, you know, essentially evolutionarily, all of these animals have evolved the same response to hypoxia, but independently, as they've gone to these hypoxic environments, they have evolved a mutation, a similar mutation, not the same, independently evolved a similar mutation in the same gene to modulate the same pathway, right? This is convergent evolution, right? And is a pretty good story. Okay, so if we look at all of the species that have show a signal for EPAS-1 in these GWAS studies, we've got Denisovians. They're extinct, but they're still in the list. You got a Tibetan gray wolf, you got a Tibetan horse, an Andean horse, a Tibetan sheep, yak, Tibetan pigs, Tibetan wild boars, goats, you know, lots of stuff. Bunch of fowl, a couple of rodents, and a snake that have all evolved a similar response to this solution. But there are other animals, specifically Tibetan humans and Tibetan dogs, that also that if you look at their EPAS-1 genes, you can find a similar response in those genes to what you see everywhere else. But it didn't evolve in either humans or dogs. Well, how did it get there, you ask? Turns out that the copy of the EPAS-1 locus that was identified in this GWAS study is nearly identical to the one found in Denisovians. The EPAS-1 locus that shows up in the GWAS study for the Tibetan dog is nearly identical to the one that shows up in the Tibetan wolf. So what happened is it evolved, so the EPAS-1 response evolved in Denisovians, evolved in wolves, and then at some point, dogs crossed with wolves, humans crossed with Denisovans, and that well-adapted allele came into those populations and was able to propagate, right? So adaptive introgression, all right, as a mechanism to improve, so it's clear evidence of adaptive, the ability of adaptive introgression to improve a species' ability to adapt. Okay, now this is an image that you guys are going to be looking at, or a similar image that you guys are going to be looking at a lot. It's the integrative genomics viewer, and what you do with it, and what I'll be talking about with it in this presentation are single nucleotide polymorphism variants and haplotypes. Okay, so when you look at something like this, each one of these rows represents sequence data from a different animal, and everything that is colored, and it's mapped to these animals are mapped to the equine reference, and the horizontal bars are sequencing, high throughput sequencing reads. The vertical bars indicate the number of reads that span that particular base position, and they are colored based on whether or not the allele at that position agrees with the reference, right? So everywhere that you see gray, the animal at that position agrees with the reference. Everywhere that you see a different color, and the key is over here. A's are green, C's are blue, G's are brown, and T's are red. Tells you that that animal has an allele of that character at that position. And so everywhere here you see in the wiggle plot, you see solid colored bars, at least for the first three animals, and this one on the bottom. what that's telling you is that that animal is homozygous at those positions. For this one particularly for a T. This animal is heterozygous across all of this, across this region. And you can see what the two alleles are based on the two colors that are present at that position. Now, when I talk about what is inherited and what is passed and what is introgressed, it is always at the level of a haplotype, right? Typically, when we look at genomes and genome sequences, we look at this coalesced version of the two haplotypes, right? And they're not phased. There are sequencing technologies that can dephase these things, and they're really good, but they're expensive, and they were beyond the scope of what we were doing here. But phasing is an operation that can be done if you've got enough data relatively easily by eye, and there are algorithms that do it more or less the same way that I'm going to show you how to do it here. So when you've got homozygous variation across a region, what you can know for certain is that that individual has two copies of the same haplotype, right? So these three animals each have two copies of an identical haplotype. This animal, even though you see no variation, it's just consistent, completely consistent with the reference. It carries two haplotypes that are identical with the reference genome. This animal carries two haplotypes that are identical. This animal is heterozygous. There are 8 polymorphisms here, right? And there are 2 to the 8th different haplotypes that you could imagine that could create that pattern of heterozygosity. But this animal's only got 2, right? So how do we figure out which they are? It's a relatively simple process if you know what the common haplotypes are, because when you see heterozygosity like this, you can ask yourself, is this pattern of heterozygosity recapitulated if I simply convolve 2 of the common haplotypes that I've got? And in this case, it is. This individual has a copy of this haplotype, and it has a copy of this haplotype, producing this this pattern. So most everything that we're going to be talking about, again, is going to be happening at the haplotype level. Okay, so back to introgression. Is there evidence for this in the horse? Doctors Bailey and Balasuria worked on a project where they identified an allele in the horse that conferred susceptibility for persistent shedder status of equine artervirus. And what Dr. Bailey found when he was looking at this, and I've labeled this image of IGV, where up these first 3 animals, one is a donkey, one is a Grevy zebra, one is an onager. The bottom 3, 2 thoroughbreds here in a row, and a saddlebred. What they noticed is, so this is in fact the haplotype that confers susceptibility. And he noticed that of all of the haplotypes that he looked for in this region, that haplotype was similar to none of them in horses, right? It just sort of existed all by itself. You had a couple of different, other different haplotypes, but given that this is a 240 base pair region and there are 8 polymorphisms in it, that tells the well-trained eye that these are 2 haplotypes that are evolved from one another by several million years. You just can't see that much fixed difference between 2 haplotypes without that much time to change them, right? And in all of the horses that he looked at, there was nothing in the middle. There was nothing to suggest that there was a path that one could get to haplotype A from the rest of the haplotypes in the horse. So he looked at other non-cabalene equids, the donkey, the Grevy, and the onager, and lo and behold, the haplotype the haplotypes that you see there are nearly identical to the susceptibility haplotype that they found in the horse, right? That's a big deal, right? It looks very much like this haplotype came across in an introgression event, That is the explanation that that seems to make the most sense. Okay, so given that, we came up with a hypothesis. So if there was an introgression event, then there must be more of these that can be found. If they're still there, they must be functional. Our question, can we find them? And then there's the obvious elephant in the room. Well, among extant equids, you can't hybridize a horse productively. Okay, again, that's something that I hope to address by suggesting that these events, so given the number of variants differing between the non-cab and the cab, these appear to be events that would have had to have happened millions of years ago to have accumulated that much difference between the two species. If it happened millions of years ago, there's conceivably another non-cabaline equid that existed that had these alleles that could have interbred, and that's more or less what we're guessing happened. Okay, so how do we know that any given region would have come from a different species? So I'll throw up this phylogenetic tree again. You all are used to looking at these. Typically, these are derived. Historically, these were derived from fossil records, right? But now that we have these wonderful genomes, and now that we know of all of the genetic variation that exists between these species, right? We can derive a phylogenetic tree based on drift at the nucleotide level in a genome. Okay. Now, if we were to take a horse genome in its entirety, and a donkey genome, and a grevy, and build phylogenetic trees based on what we see there, right? It would recapitulate what was derived from the fossil record almost exactly. Right? But different regions of the genome are going to evolve at different rates, right? And we can look at different regions of the genome and begin to ask questions like, is this particular region well conserved in this species, or is it one that's hypervariable, or are there two haplotypes that show up in here that really that you can't make a plausible argument that they came from the same place recently. Okay, so back to this picture. Again, Dr. Bailey looked at this, and what the way that we approached the problem was to phase the haplotypes that we find here, and then plug them into an algorithm and endeavor to derive a phylogenetic tree. And this is what we get. Right? We find two cabaline haplotypes that were derived from the 14 horses that we looked at that look nothing at all like the non-cab haplotypes. Yet within those 14 horses, there existed one haplotype that clusters in the same clade with the haplotypes from the non-cabs. So this is the signature that we are going to be looking for. Right? So you've got a haplotype. All right, we'll go back here. We've got this haplotype. And if you were endeavoring to do an evolutionary phylogenetic analysis, does it look more consistent with an interspecies transfer than it does if it were to have evolved from other haplotypes that occur within that species? And the phylogenetic tree that we were able to derive from that suggests that yes, in fact, it does. Okay, so let's talk. So when we're looking at these alleles, there's a concept that you need to understand, and that is the concept of an ancestral allele. Okay, so this is just a simplification of that phylogenetic tree that you were just looking at. Let's imagine that once upon a time, there was a most recent common ancestor to all of these animals. And it existed several million years ago, and at some position in the genome, it had an A. And then horses split from the non-cabalines some 4 million years ago. And then some 2 million years after that, the asses split from the zebras. But all along, the A allele that was common in the most recent common ancestor was conserved and retained by all of these species. Okay, but the horse derived a new allele, right? A new allele appeared in the horse and it became fixed and it's a G. Okay, this G we're going to call a species-specific allele, right? So if I look at an animal and I see a G here, I can be pretty certain that it's a horse because it doesn't occur at high frequency. It doesn't occur at all or at high frequency in any of these other animals. Okay, so if I also see that ancestral A among horses, right? Well, there are two different ways that it could get there. One is simply to follow the path, right? The horse evolved a G and there wasn't a complete conversion of all of the species-wide of all of the A's to a G. And it's just there at some nominal frequency. Another possibility is that A was introgressed, right? That there was some introgression event at some point in evolutionary time where that A was brought back into the horse lineage, right? So two different possibilities for that A to get back into the horse. Okay, so when we're looking at IGV, we look at our donkey grevy and our onager, and what we see is that this G is almost certainly the ancestral allele. The most recent common ancestor almost certainly had a G, and that G can also be found in horses, right? Along with a derived allele, which is an A, right? So you've also got, and the allele that shows up in the reference is an A. At this position, just 2 nucleotides over, you see another ancestral allele, which is a C, but in horses, that allele has shifted to a T and has effectively been ablated. Can't find C anywhere in horses. Okay, so those are two classes of these of alleles that we're going to be looking at. Okay, so we wanted to look for introgression signatures in horse DNA. So Dr. Bailey, looking at a 240 base pair window, he could probably be done with it in about a minute. And if he's scanning across a 2.6 gigabase genome and we extrapolate, it's going to take him about 20 years to do that. And then he can move on to the next horse. So he doesn't want to do that. So what was our solution? We need to develop an algorithm. He came to me, we need to develop an algorithm. And I said, I know, but I have no idea how to do it. So months went by, I'm thinking about this. And this is what I finally came up with. Okay, so this is an expanded region, and I cherry picked a bit. So the region that's circled here, it is a putatively introgressed region in the horse. Okay, so looking at this just by eye, what do you notice? The donkey, the grevy, and the onager, the SNP distribution there is pretty consistent, right? When one of those species has a variant, the other species, tend to line right up with it. That's pretty cool. So they are these species-specific variants that we were talking about, right? Where the horse has moved away from all of them. But they're very dense, right? Lots of color up there. Whereas if we look at the horse, right, and this is like 24 kb. If we look at the horse, we don't see a whole lot of polymorphism, right? Most of the time, not a lot of density. But if we look at this region, right, we see a great deal of density, right? So the notion for the development of this algorithm was, can we do something based on the density of these species-specific alleles that can clue us into whether or not a particular region is likely to have come over from another species. So that was exciting when we had that idea. And I leapt immediately to the notion, yes?
So when you're comparing it to the horse reference.
Yeah.
So it's the density of variance relative to that.
Correct. This was exciting because this was something that I dealt with back as a physical chemist, and I got to go back and I got to look at the physics literature, and the good folks who do physics, they helped us out. So they had worked just on a simple formula that explained a neighbor distance function for systems of interacting particles. And if we think of those systems of interacting particles just as SNPs, there are 11, so I've rendered along the top 11 SNPs with 10 distances between them, right? If there's a greater density of SNPs within a region, right, those distances are going to be smaller. If there's a lower density, those distances are going to be larger. So if you created a histogram of all of those distances, you know, might that look different in for a more dense system than a less dense system? So the folks at who wrote this paper came up with this formula that explains it well. It's a simple decaying exponential that's essentially dependent upon the density, right? The more dense it is, the more rapidly it decays, and what that's effectively saying is the more dense, the more likely it is that you're going to find a neighbor more close, right? Pretty simple concept. Okay, so we don't, with these SNPs, they're not non-interacting particles. There are lots of processes going on, so we can't fit it to a simple function, but we can derive these nearest neighbor distribution functions empirically. And so essentially what we do is we go through the genome and we look for these species-specific differences, right? Where the horse and all horses have one allele and the non-calves have another, right? And we create that distribution function, and what that is what you see here in blue, right? It's more dense, it decays pretty rapidly. I can get that to go away, can't I? Oh, that was bad. I didn't want to do that. Right there? Okay. Can I move it?
Yeah.
Okay. Okay, so the horse genome has about 2.6 million or 2.6 billion bases. Horse-specific alleles, that is the number of alleles that distinguish a horse from these non-calves, about 1% of the genome. So 99%, horses are 99% similar to all of those other things. Ancestral alleles, so of these 22 million, you can find in horses about 4 million of those ancestral alleles, right? Where a horse has evolved a new allele but hasn't completely obliterated the old allele, right? And if you plot the distribution function of where you can find these ancestral alleles and where you find these horse-specific alleles, the horse-specific alleles, it's more dense, more rapid decay. The ancestral alleles are a longer decay, longer tail. All right, so long story short, if you find two SNPs that are separated by 100 base pairs, it's more likely from, more likely consistent with an introgressed allele. And if you find something that's out at about 300, it's more likely something that's just an ancestral allele that came down along the normal evolutionary path. But you're not going to, but we don't think about these things SNP by SNP. We think about them by region, right? Okay, so how would we evaluate this for a region, right? So the algorithm that we essentially came up with was to come up with a maximum likelihood estimate. And so the maximum likelihood estimate, what we looked at was the product of all of these distances within a given region. So we took 20 polymorphisms, right? So starting here, the next 20 polymorphisms, and we calculated this maximum likelihood estimate by calculating the ratio of the probability that it was from the horse-derived alleles or from the ancestral alleles for every the product going all the way up to 20 SNPs out. So we calculated it for just the 1 SNP, for the first pair of SNPs, for 3 SNPs, and essentially we asked ourselves, okay, what's the maximum value that's in there? And that, you know, the biggest one, that's the one that we're going to evaluate for signal, right? So if I get a big value, so if you've got a value that's very, very large, right, then it's most likely that the distribution that those SNPs came from is the are the horse-specific alleles, and if it's very low, then the distribution that those SNPs came from is most likely the ancestral path. Okay, so MLEs, and this 100 number was derived by trial and error, but I'll show you later how it is that we came to justify it. Okay, so example of a negative result. Okay, within here you can see we can see some polymorphisms that, right? So a species-specific polymorphism here. There's one here. There's one here. Here there are about 8 of them here. 8 of them here, giving us 7 distances. So we derive our probability from those current functions, from the horse-specific distribution, and then from the ancestral distribution, and we divide one by the other, and we get a maximum likelihood estimate of 0.63. So we only looked at those. We're only looking at those SNPs, right, that are species specific, where the horse derives something different, and the animals, the non-cabalene, are fixed for something else. So we only looked at that group of polymorphisms. And these are just two spread out. It's not a good candidate. And if you look closely at all of the SNPs that are in the thoroughbreds and the saddlebred, right? There's a lot of change, and they don't look terribly like what you see in the non-calves. So, you know, the algorithm is giving us good guidance. Now, if we look at, again, this is Dr. Bailey's result, and we've got, I think, 8 SNPs and 7 distances. You know, the first pair of SNPs separated by 21 bases. The next SNP is separated. Next 2 SNPs are separated by 138, 129. You plug all of that in, and you get a maximum likelihood estimate of 403. Right? So that's one we probably want to pay attention to. Okay, so we take, again, we took, if we take the haplotypes that you pull out of Dr. Bailey's, the region that he identified, build the phylogenetic tree, it shows you exactly what we would expect to see, right? The cabalene haplotypes grouping cleanly away from the non-cabalene, but evidence of a cabalene haplotype clustering in the non-cab clade. Okay, so another positive result. This is a much shorter distance. We get a phylogenetic tree that's going to look something like this. So there's typically not a lot of variation on the horse side in these regions, and the outlier in the cabalene, again, clustering with the non-cabs. Okay, so what animals did we use in this analysis? Right, for non-cabalene equids, we used a total of 9 animals: 2 donkeys, 1 Somali ass, 4 zebras, a king, and an onager. Horses, we used 2 saddlebreds, 7 thoroughbreds, 1 of which was Twilight, a halflinger, 3 Arabians, 1 standardbred. And why did I pick these animals? I could get the data for free. So that's largely why they're in this list. Okay, so when we run that algorithm over those animals, what do we find? So standardbreds, halflinger, Arabians, all between 4,000 and 5,000, you know, on the low side of 4,000 of these putatively introgressed regions in each animal. The thoroughbreds are all pretty consistently lower, all right, between 2,000 and 3,000 variants. Why is this? All right, and Twilight, you'll notice, is 0. Why is twilight 0? Well, one of the qualifications that we had when we looked for a haplotype, we didn't want any guesswork to be done in the phasing. The animal had to be homozygous for a particular haplotype in order for us to consider it, right? Twilight can't homozygously vary from herself, so she would have had absolutely no signal across the region. And that is most likely why the remainder of the thoroughbreds are low, right? So what this shows you are the introgressed regions that the twilight does not have. And odds are those other thoroughbreds share introgressed regions that are simply invisible because she has them. So if we get another reference genome from another breed, those numbers will likely change. Okay, so what do? We took all of those, we coalesced them, the ones that overlapped, and we get a genome-wide signal that looks something like this, with signals as high as 10 to the 8th. Big peak over there at chromosome 20, that's MHC, which got us really excited, right? So MHC has an adaptive response, right? If you're challenged with something, MHC is going to react to that. And consistent with the story that I presented early on, if a new species comes into the environment and it's ill-equipped to deal with some onslaught, crossing with one of these non-cabalines that had adapted a response to viruses, whatever, and bringing all of those alleles in very quickly would have been a huge advantage, right? But we've got in total 15,583 regions that had a maximum likelihood estimate of greater than 100, spanning about 1.1% of the genome. Okay, so how do we evaluate a predicted region? So great, Ted, you've got 15,000 of these. How do you know they're right? For a predicted region, what we would do is we would phase the haplotypes for all the animals using all the variation. So previously, when we were looking for regions, we just used those horse-specific alleles, right? Now we use all the variation that's present in all the animals. We derive haplotypes. We phase haplotypes. We label a haplotype by origin, either cabaline or non-cabaline. We collapse to a unique set within the cabalines and within the non-cabs, and then we construct a phylogenetic tree with Mr. Bayes. Assuming that we have the two clades, cab and non-cab, a positive will be if a cab haplotype is found in the non-cab clade. Negative or false positive will be if no cab haplotype is found in the cab clade, or in the non-cab clade, sorry. Okay, so a positive, this is lovely. We see these two cabalines clustering with all the non-cabs, and then two other cabaline haplotypes separated well away from them. Okay, false positive. Here there were 6 cabaline, is that 5 or 6? 5 cabaline haplotypes. They all clustered together and well away from the non-cabs. No cabalines in here, right? So our algorithm identified this region as possible. It's not what we're looking for. Okay, so we wrote some code to look at the markup language that creates this thing and ask the question: how far across the phylogenetic tree do I have to go in order to get to a bar, or in order to get to a bar that puts me near calves, right? In order to get to non-calves rather. So in order to get to the non-calves, I've got to go to the topmost node here. So that's not good, right? That's not a positive signal. If we look at this one, for these two haplotypes, you have to go all the way to the origin node. For these two cab haplotypes, you only have to go about a third of the way up before you have the bar that defines the clade of non-cabs. So this is a legitimate positive. So we ran that algorithm over all of the phylogenetic trees that we had generated, and we get a curve that looks something like this, right? Below the x-axis, you have the number of cabalene haplotypes that were found in the non-cab clade. Okay, all the way over on the left is 0, right? About 25% of the regions that we found did not have a cabalene haplotype that clustered with the non-cabs, right? False positives. So we've got about a 25% false positive rate. If I set that maximum likelihood threshold much below 100, that number begins to go way up. The ones that we're really excited about are the ones with 1, 2, and 3 cab haplotypes in the non-cab clade. The ones with higher numbers are a little more difficult for us to understand or explain, right? Because that's a lot more variation than we're seeing in these putatively introgressed haplotypes. Typically, they come over and they stay pretty much unchanged. Okay, so what's the biological significance? We are still working on that. The strong signal at the MHC region is exciting, in as much as it's known for its adaptive response. We're also going to be looking at T cells and immunoglobulin. Otherwise, the functional annotation that we've got right now for the equine genome is a bit lacking to assess this well, right? Exons are annotated. You've got some annotation of methylation regions, but what regions are functional and how are wanting. Very few of these, the one that Ernie had identified, did in fact overlap with an exon. What I think that we're going to see is that most of these are going to be overlapping with regulatory regions, and the phenotypic impact that we're going to see is going to be in allele-specific expression. Okay, next steps. We want to sequence a lot more non-cabalene equids to get a more accurate picture of horse-derived alleles versus ancient. We need to generate more equine reference quality genomes to remove the bias that's introduced by twilight. We've got an active effort underway to generate phage genomes for a Shire crossed with an Arabian. That's work that I'm doing with Siobhan Labs and Jessica Peterson. This is going to give us the opportunity to look at low-frequency alleles and generate more FANG-like data. So those of you not as familiar with the FANG project, for an animal that is in it, you sequence the whole genome sequence as well as RNA-seq data for a whole litany of tissues. Being able to have the whole genome sequence data to assess potential introgressed regions, as well as expression data for proximal genes, is going to allow us to do the association studies that one would have to do in order to verify the impact of some of these regions. Okay, so I'll wrap up quickly. For applications, these things, if they are still there, right? If they have not been driven out of the horse population, and they are all one of the I think I said earlier, when I was looking for these regions, the animal in which we were looking had to be homozygous for these haplotypes, right? That means that these are high frequency haplotypes, right? So they're there and they're high frequency. If they're still there, they have to be important. In the wild, evidence suggests that introgression is an important evolutionary mechanism that can quickly distribute adaptive genes. And the notion of species diversity, right? So for many agricultural species, this is an impossibility. But to be able to have closely related species with which you can make a viable cross to bring in well-adapted genes would be is something that is essential to maintaining ecological health. Okay, so acknowledgements, collaborators, of course, did a lot of this with Ernie Bailey, Dr. Siobhan Laux, used a couple of Naveed's horses, and Dr. David Fardo from next door at the medical campus was invaluable in discussing the algorithm, which he called, and this is the highest praise I've ever gotten from a statistician, completely defensible. So at U of L, Kalpani De Silva, a graduate student of mine, she's helped with this. The Fang and the Fang horses, Dr. Jessica Peterson, Dr. Carrie Fino, Dr. Rebecca Ballone. The EPAS-1 work, Dr. Peter Hackett gave me a lot of insight on that. And for funding, the bioinformatics fund from the Janet Kohler gift. With that, I am happy to take any questions. Yes, sir.
First off, Dr. Adam wanted to remind you to repeat the question for the benefit of those on Zoom. Questions asked from here. Repeat the question.
Oh.
And I'll ask the first question.
Okay.
Kind of a two-part question. One, in simple terms, you said very few of the regions appear to be introgressed, overlapping with exons.
Right.
Which is surprising to me. What specifically, what is a few? What, 20% or something like that?
It's around 5%.
5%.
Yeah.
So a bigger question is, why do you think that? I think you mentioned regulatory regions, but
I don't know. The first thing that we expected to see an enrichment around exons. We given that that's how it was found, the SNPs that Ernie found, they in fact changed amino acids, right? It just, we were certain that we were going to find enrichment there. It just didn't exist. Why is that? I don't know. You can find it, I wish that I had an answer. And I didn't repeat the question just after you told me to do that. Yeah. And Dr. Howe's question was, why did we find so few of these regions overlapping exons, and why do I think that is? Yes.
So in your events that look like true positives, did they sort among the non-cavalines into zebras like your title suggests, or were there just as many in asses and onagers, and is it just non-cavalines in general, and, or were they overrepresented in the zebra genome? And then related to that, can you circle back to when they had the same number of chromosomes, and was that before there were zebras and asses? So there's really no way to know the answer to that.
Yeah, so repeat the question. Thank you. All right. So did the zebra, in the phylogenetic trees, did the zebras and the asses all cluster together?
No, in your positive.
In the positives?
I think this is an introgression event. Your green things,
Yeah.
1, 2, and 3. If you then parse them into what non-cav genome did they align with, were the zebras way overrepresented? And like horse and zebras sort of liked each other's, but the horse and the onagers didn't. So it really looked like an evolution, way back when horse-zebra party happened. And so that's where these introgressions are happening, or could it have come from any of the non-cavaline species?
Okay, so is there evidence in the phylogenetic tree that the haplotypes came from one particular non-cavaline, like the zebras or the asses, or did it just come from a non-cavaline? So right now the evidence points to a non-cavaline. It doesn't consistently group with zebras or the African or the Asian asses. There's a lot of haplotypic diversity there, and it's inconsistent which one the cabalene is most close to. I used zebra because I had to pick an animal, and we were worried that ass might be pejorative. Yeah, we don't know. And yes, the introgression event could have been before those animals diverged. Absolutely. Anything?
Okay, I'll
Yes.
start over. So those ones with your wonderful density map that turn out to be false positives, how do you explain those? Like, how did that unlikelihood happen where you have all this variation in these real high ratios, and then it doesn't make sense because it's not in the non-cat?
These are all just probabilities. Yeah. So yeah. So if
You have to repeat the question.
right. So in the false positives, what went wrong there? Right. Why did I identify that as a putatively introgressed region, and ultimately it was not? Right.
How do you explain it?
How do I explain why that would be? And my answer in short was these are all just probabilities, and we expect to miss a couple of times. So if you've got a number of SNPs that are so that where those two curves cross over one another is at about 180 nucleotides. So if you've got 4 polymorphisms that came, evolutionarily like you'd expect down the horse line, and they're spaced at 120 nucleotides apart from one another, that's going to give a signal. And yeah, so but for deriving the phylogenetic tree from the haplotypes that are there, you wouldn't have known otherwise. Yeah. What do I think the proportion of false negatives would be? What a great question, and it was so easy to repeat. Yeah. Right. So looking at what we're missing from Twilight, right? I mean, so there are easily a couple thousand out there. Yeah, where they are and what they're doing. Maybe those are the ones that are sitting on top of the exons. Who knows? But yeah, I think that we're certainly missing thousands. Whether it's tens of thousands, I doubt it. But yeah. Anything on chat?
All right. Thanks very much, Dave.
My pleasure. Thank you.